All right, guys, hi. So, two more tough topics to get through: photosynthesis and cellular respiration. Photosynthesis, the plants are taking in sunlight and carbon dioxide and producing oxygen and sugars. And then respiration is where you utilize those sugars through a chemical reaction with oxygen. So, the goal of photosynthesis is to produce sugars using the energy of the sun. Carbon dioxide and water in the presence of sunlight go to glucose and oxygen. And you guys have seen this diagram in your freshman year. We are utilizing light in this process. Light is a form of energy, and realize that the white light that you see from the sun or from your lamp actually contains all of the colors. The colors are represented by wavelengths, and the wavelengths have different amounts of energy in them. So each color is a different wavelength and represents a different amount of energy. Violet, being the shortest wavelength, has the most amount of energy to it. Red, being the longest wavelength, has the least amount of energy. We have chlorophyll as the main photosynthetic pigment. There are accessory pigments in there, but chlorophyll is the one that does the bulk of the work. This is found in the chloroplast of the cell, and it causes the green color of the plants. And your plants are green because the chlorophyll reflects the green light. So the color that you perceive is actually the color that's being reflected by something to your eyes. So my shirt, if it wasn't like this dark cave in our office, would be green, which means it's reflecting green to your eyes. The top bar of this slide is yellow, which means it's reflecting the yellow to your eyes and absorbing all other colors. Several different kinds exist, each specializing in a specific absorption spectrum. The two that we most focus on are chlorophyll A and chlorophyll B. Here is our light absorption by chlorophyll, and you see that it peaks in that 425 range, so between 400 and 450, and then it peaks again between 650 and about 700. There is a slight difference between the action spectrum and the absorption spectrum. So the absorption spectrum is all of the light that the plant can absorb. The action spectrum is all of the light that is actually being used and converting our water and our carbon dioxide into sugar. Note that the lines are almost identical with the exception of this little area right here. The one divergence is an area of the spectrum utilized by the accessory pigments. So how do we use light energy? We do it in the light dependent stage. Light dependent, wow, that is an old term, but it just means that you need light to do this. So light energy is used to split the water molecules. That process of splitting water molecules is called photolysis, and we split them into hydrogen ions and oxygen. That oxygen is the byproduct, and we release it out, and that's what allows you to be alive. Those hydrogen ions are then sent through a series of enzyme-driven reactions that then produce some ATP. The hydrogen ATP created in the light-dependent reaction are used to fix carbon dioxide in the light-independent reaction. And so this top bar is your light-dependent reaction. So you have 12 waters plus light going to 24 hydrogen ions, 24 electrons, which then go to 18 ATP and 12 NADPH. This ATP and NADPH are then utilized in the light-independent reaction with carbon dioxide to make sugar and water. Here it is again in just another pictorial form. So how are we going to measure the rate of photosynthesis? We can measure the rate at which the reactants are used or the rate at which the products are produced. So our reactant here is carbon dioxide. We can actually measure how fast the plant is intaking carbon dioxide. Obviously, the faster the plant takes in carbon dioxide, the more photosynthesis is being done. We can measure oxygen production, and you guys, I think, did that in AP Bio, if you were there, where you saw the bubbles coming up off the plant. We can measure the amount of glucose produced. So the weight of the plant will change as glucose is produced. The plant needs to be dehydrated to ensure that it's not just retaining water. We all have bad months where we bloat and retain water. So we're going to take our plant, we're going to dehydrate it, and then if there's a added weight, it's purely the sugar. Light is a limiting factor. Without light, the plants will quickly utilize all of the remaining reactants needed for the light-independent reaction. At this point, the plant starts respiration. Oh my gosh, plants do respiration. So the plants need light in order to do photosynthesis. And if we don't give it light, it's going to do the last little bit of photosynthesis that it can, and then it still needs to live. And so it starts breaking down the sugars that it was storing. So it starts doing respiration. If light's available, then the light-dependent reaction takes place. Eventually, additional light will not produce additional light-dependent reactions. Remember, I said that the light-dependent reactions are all enzyme-based. If your enzymes are all busy, it doesn't matter how much more light you throw at that plant, it's not going to do more photosynthesis. And we have the idea of a compensation point. This is the point where photosynthetic production of oxygen is equal to the oxygen used by the plant. So right here, the plant is doing as much photosynthesis as it is doing respiration. That's your compensation point. Carbon dioxide is a limiting factor here. It slows the light-independent reactions. NADPH will accumulate in the stroma, and the concentration of NADP reduces. Now, if we have no acceptor for that hydrogen plus, the photosystems in non-cyclic photophosphorylation back up. By the way, this term photophosphorylation is a scary term. Photo means light, and phosphorylation is essentially the production of ATP. So if we're doing photophosphorylation, we're using light to make ATP. The cells will go into cyclic photophosphorylation. The excited electrons in photosystem 1 fall back into their original position, and the hydrogen ions are transferred to the thylakoid space, and you get some ATP. Cyclic photophosphorylation is that last-ditch effort for this plant to continue doing photosynthesis. It's not a long-term solution. Temperature is a limiting factor. I said this was all enzyme driven, and if we go above the ideal temperature range, those enzymes break down, in which case we cannot do photosynthesis. If it's too cold, the enzymes are still functional, but the molecules aren't bouncing into those enzymes in a fast enough manner to do photosynthesis. The light-dependent independent reactions are largely unaffected. All right, so here are some graphs. As the concentration of carbon dioxide increases, the rate of photosynthesis increases until a point, at which point all the enzymes are busy. Temperature, again, is that perfect enzyme kind of curve that you're used to seeing. The limiting factor is the thing that will prevent the plant from doing photosynthesis first. So the highest rate of photosynthesis is produced by providing a plant with its optimum temperature, maximum light intensity, and maximum carbon dioxide. The factor furthest from optimum becomes the limiting factor. All right, we're going to pause for a second, but no, even if you're SL, because my classes run like an HL, you'll still need to know this stuff. All right, moving on to the higher level material. Now, for all of my exams, you need to know both the SL and the HL, but when you go to sit the final exam, the majority of you only need the SL material. Here's the deal though, it never hurts you to be too smart. I would rather give you all of this information now and have you learn it than have you learn just the bare minimum. Bozeman biology, please watch this. It goes into it in great depth and it will help your understanding. So the first thing here is you need to be able to label a micrograph of a chloroplast. So you need to know where all of these things are, the grana, the thylakoid, the stroma, the chloroplast membranes, inner and outer, starch granules. The good news on this is it'll be a 3 or 4 point question on probably paper 2, maybe a 1 point multiple choice question. So make sure that you can identify 4 of these things and you'll probably be okay. You need to know the structure of the chloroplast and its role in photosynthesis. So the double membrane bounding the chloroplast contains the grana and the strona, and it's permeable, which means it allows in the carbon dioxide, oxygen, ATP, and sugars. The 2 photosystems, and there are 2, photosystem 1 and 2, on the thylakoid membrane are where light's absorbed. The thylakoid spaces are where we accumulate the proteins, and then they flow through ATP synthase to generate ATP. And the fluid of the stroma has all of the enzymes for the light-independent reactions. So light-dependent reactions, light energy is captured by the chlorophyll. This energy is then used to split water. Chlorophyll molecules are grouped together in the thylakoid membranes on the grana in photosystems. Contains several hundred chlorophyll molecules and some accessory pigments. The accessory pigments allow it to pick up excess colors, excess energy, excess wavelengths of light. There are two photosystems, and they were named in order of discovery, not in order of use. So yay bio, right? So photosystem 2 is also known as P680 because it absorbs the 680 nanometer wavelength of light. Photosystem 1 is the second one used, and it's also known as P700 because it activates or absorbs the wavelength of 700 nanometers. When light enters the photosystems, the ground state electrons are raised to an excited state. And so if they're down here, they have very little energy. And light hits it and they bounce up to a higher level. And now they've got all of this potential energy that they release as they go back down to ground level. So these excited electrons leave the chlorophyll molecule and are replaced by new ground state electrons. Here it is in pictorial form. Make sure you take a second to look at this diagram. All right, photosystem 2. First, the excited electrons from photosystem 2 are picked up and passed along a chain of electron carriers. These are all embedded enzymes. Some of the energy from these electron hydrogen ions are pumped across the chloroplast matrix to the thylakoid spaces. This produces a little bit of ATP. The energy lost for ATP production drops some of these electrons back to the ground state. These fill the vacancy left in photosystem 1, and the electrons have now been passed from photosystem 2 to photosystem 1. Photosystem 2 gets new electrons from photolysis, from the splitting of water. All right, photosystem 1. Watch the little video, guys. I'd show it, but I get copyrighted. These electrons from photosystem 1 are picked up by electron acceptors. 2 at a time, they're passed to NADP with additional hydrogen ions from photolysis, and they make NADPH. So photophosphorylation, I said it in the earlier part of the lecture, it's a big scary word, and IB likes to use the big scary words, but photo means light and phosphorylation means adding phosphorus or phosphate. And so this is the making of ATP using light. So it's linked to the electron transport via the movement of the hydrogen ions. The hydrogen ions are trapped in the thylakoid space and they flow out through ATP synthase. This is like a turbine in a dam. And as this turbine spins, it produces ATP. So it's going to turn the ADP into ATP, and it's considered non-cyclic photophosphorylation because it's a linear movement. All right, let's go through this for a second, if I can go back. So photosystem 2 is the first photosystem. Light hits photosystem 2. We have photolysis. The oxygen gets released. It's a byproduct. And the happy electrons, which we denote as hydrogens, go up to a higher level. And they're up here in the upper, and then they're being used to make ATP. And as they're making ATP, they lose energy and they're back down to the ground level. They're back down to the ground level at photosystem 1. And light will strike photosystem 1, and these electrons get happy again, and they're up here and they're losing energy and they're losing energy and they lose energy. They're losing the energy to NADP to create NADPH. So at the end of this process, you're left with ATP and NADPH. Both of these are then used in the light independent reaction. Okay, so chemiosynthesis. Chemiosynthesis is the making of a chemical. So in photosynthesis, all of this is occurring in the thylakoid space, whereas in cellular respiration, this is occurring in the mitochondria in the inner and outer membranes. In photosynthesis, the water molecules are where the proteins come from. In cellular respiration, it's from reduced hydrogen acceptors. In chemiosynthesis and photosynthesis, sunlight is the energy source, whereas in respiration, it's glucose. And in photosynthesis, the ATP diffuses into the stroma and is utilized to make sugar, whereas in cellular respiration, it's used for metabolic and other reactions like your muscles twitching. The light independent reaction now is taking our ATP and our NADPH and utilizing it to make sugar. So any ATP that's not being used by the plant at that exact moment then gets turned into sugar in the light independent reaction. Carbon dioxide is converted to a carbohydrate at this point. It occurs in the stroma of the chloroplast, which is an area surrounding the grana. And the first step is fixation. So carbon dioxide is fixed into glycerate 3-phosphate. Then glycerate 3-phosphate is reduced into triose phosphate. This reduction phase uses a little bit of NADPH and a little bit of ATP. Then our product is synthesized. Well, what's our product? It's glucose. So our triose phosphate is metabolized to produce carbohydrates. Sorry, guys. We're using carbon dioxide to produce carbohydrates. And then because this is a cycle, right, we need to use anything that's left over and we need to regenerate it so that it can start the cycle again. So some of the triose phosphate is metabolized to produce the molecule that reacts initially at the beginning of the cycle. This cheesy Calvin cycle song is totally worth a listen. All right, so we fix, and then we reduce, and then we product synthesis, and then we regenerate. And then we fix, we reduce, product synthesis, regenerate. Hank, guys, no good lecture ends without a little Hank. So please watch the Bozeman and the Hank because you really do need to understand this. And the more voices you can hear this from, the better. And then in your study groups, guys, talk to each other about it because the best way you're going to understand all of these complicated processes is to vocalize it.